Thank you for choosing American Pest CEUs.
Hi, I'm Sean Reeves with American Pest CEUs. I've been in the pest management industry since 1999. I'm an associate certified entomologist, and I'll be your instructor for today's course. Today's course was compiled by Jack Launius, who has served in the pest management industry since 1990. Our course today is going to be on mosquito, bionomics, recognition, and inspections. Bionomics is the study of the mode of life of organisms in their natural habitat and their adaptations to their surroundings, ecology. Mosquito identification is one of the crucial steps in identifying risk for both biting mosquitoes and mosquito-borne diseases in an area. Before conducting any mosquito control, you should learn more about the biology of the different mosquitoes you are trying to control. Morphological traits of mosquitoes can often be very similar. However, appearance, as well as behavior, can vary greatly from species to species. Differences in appearance, especially in the larval and adult stages, allow accurate identification of the species. Behavioral differences permit various species to occupy numerous ecological niches with relatively little overlapping. Thus, knowledge of the source or breeding habitat of mosquitoes can provide a strong clue to their identification. While many of the physical differences between species are minor and require specialists to fully comprehend, many others are readily apparent, even to workers with limited training and experience. Fortunately, most of the important Louisiana species are easily identifiable, often by a single observed feature. Many of the breeding sites for these important species are easily recognized. This section of the course is designed to provide mosquito control workers with basic information, including some shortcuts which will equip them to deal with the important and most common mosquitoes more effectively. The following covers the bionomics of each species and is a list of the principal identifying features. Selected biological and ecological data on important species of mosquitoes are summarized below. Floodwater mosquitoes lay eggs singly on non-aquatic surfaces outside of containers. An example of this would be Aedes sollicitans. Tan salt marsh mosquito. Aedes sollicitans is a salt marsh breeding species and is one of the most important pest mosquitoes. It is a known vector of Venezuelan equine encephalitis (VEE) and a suspected vector of Eastern equine encephalitis (EEE). Aedes sollicitans breeds along the Gulf coastal plains and inland areas where brackish water is available. The eggs of Aedes sollicitans are laid on moist soil intermediate to freshwater marshes, where they remain until flooded by high tides or rains. Breeding may occur in potholes, depressions, and runs, small ditch-like depressions containing specific plant species, in otherwise level areas of the marshes, which are subject to periodic but not daily flooding. The eggs require several days of drying, conditioning, in order to hatch when flooded. Development of the aquatic stages requires 7 to 10 days, depending on water temperature. The adults are strong flyers, often migrating in large swamps from the breeding areas in search of blood and host animals. They commonly fly 5 to 10 miles and may travel up to 40 miles when aided by favorable winds. Migration flights take place during early darkness, and the adults are attracted to lights. During the day, the mosquitoes rest among the vegetation, where they will readily attack any animal that disturbs them. They have made certain areas literally uninhabitable for humans and large animals. Principal identifying characteristics: 1. Medium-sized golden brown species. 2. Female abdomen is pointed. 3. Pale bands on bases of most tarsal segments. 4. Pale band on the proboscis. 5. Basal, dorsal bands on abdominal segments. 6. Medium pale stripe running the length of the abdomen. Aedes teinorhynchus, black salt marsh mosquito. Aedes teinorhynchus is not as prevalent as Ae sollicitans. Nevertheless, it is sufficiently abundant to constitute a serious problem at times in certain coastal areas and in some inland situations. The breeding habits and its biology are like those of Ae sollicitans. Although it may breed in freshwater pools near salt marshes, the adults are strong flyers and bite freely at night or in shade. Principal identifying characteristics: 1. Medium-sized to small golden dark brown species. 2. Female abdomen is pointed. 3. Narrow pale bands on the bases of most segments of the tarsi and proboscis. And 4. Basal dorsal bands on abdominal segments. Aedes vexans, inland floodwater mosquito. Aedes vexans is a floodwater and temporary rain pool breeding mosquito of paramount importance throughout Louisiana. This species causes a painful bite and is a potential vector of West Nile virus, WNV, EEE, California encephalitis, CE, and canine heartworm. Winter typically is passed in the egg stage, although extended warm winter weather can lead to off-season broods with considerable pest nuisance. Not all eggs hatch with a single flooding, and larvae appear periodically following alternate periods of flooding and drying during the season. Adults can migrate long distances from their breeding places, 10 or more miles being rather common. Principal identifying characteristics: 1. Medium-sized brown species. 2. Female abdomen is pointed. 3. Proboscis is unbanded, whereas tarsi possess very narrow, pale basal bands. 4. Basal, abdominal pale bands constricted at the middle to form an inverted V. Saurophora columbii, dark rice field mosquito. Saurophora columbii is the most widespread and important species of Saurophora in the United States. The females are fierce biters, attacking any time during the day or night in large numbers. They can kill livestock and make it practically unbearable for people to remain outdoors. For these reasons, this species would qualify as one of the most troublesome and economically important mosquitoes. Its importance was heightened during the summer of 1971 when an epidemic VEE transmission occurred in the southern United States from Mexico. Eggs of P.S. columbii are deposited on moist soil that is subject to flooding. Situations with low, rank vegetation are ideal for egg deposition. Drained rice fields, fallow rice fields, and pastures are amongst the most favorable sites. Eggs must undergo a 4 to 5 day period of maturing before being able to hatch. Once matured, they can hatch within a few minutes of being flooded, overwintering in the egg stage. The larvae of P.S. columbii are found in temporary rain pools, irrigation, and floodwaters, and in seepage ponds. The larval period is characteristically very short. During midsummer, it may be completed in as little as 3.5 to 4 days. The pupal stage requires another 1 to 2 days. The number of generations per year varies from few to many depending upon suitable hatching conditions and local temperatures. Areas which dry up and are then flooded a few days later may produce larvae with each flooding. Adults may live up to 1 to 2 months. They have a flight range of 10 miles or more. Principal identifying characteristics: 1. Medium to large black species. 2. Female abdomen is pointed. 3. Apical, pale marking on abdomen. Markings triangular or divided into paired submedian patches. 4. Proboscis broadly banded with white. 5. Tarsally basally banded with white, the first hind tarsi segment with a broad median white ring as well. Saurophora ferox, woodland mosquito. Saurophorapharix occurs in woodland areas of the Gulf Coast states. It is particularly annoying to homeowners who own homes on partially cleared woodland areas. Activity and blood feeding are most intense during daylight hours, and feeding often seems to target the upper body and head of human host, sometimes referred to as the headhunter mosquito. Principal identifying characteristics. 1. Body scales of adult are brilliant blue-purple. 2. Female abdomen pointed. 3. Last two segments of hind tarsi are totally white. 4. Larva has antenna longer than the length of head. In the photos, we're looking at container breeding mosquitoes. They lay eggs singly on non-aquatic surfaces inside of containers. And as you can see in the photo, we've got several examples of containers that could be common in people's yards. Aedes aegypti, yellow fever mosquito. Aedes aegypti is the urban vector of yellow fever, dengue fever, chikungunya, and Zika viruses throughout the tropical and subtropical regions of the world. It has also been implicated as a potential urban WMV vector. Aedes aegypti is permanently established in the southern portion of the United States, where frequently it becomes locally abundant and readily available to transmit reintroduced yellow fever or dengue viruses. Aedes aegypti is a peridomestic species of mosquito imported from Africa more than 300 years ago. In the U.S., larvae develop mainly in outdoor containers, such as tin cans, jars, bottles, dishes, and children's wading pools near human habitations. They are often found in flower vases in cemeteries, clogged roof gutters, discarded vehicle tires, and other types of human-generated artificial containers. Originally, A.E. aegypti bred in rot holes in trees and may occasionally still be found in such habitats. The adult female prefers human blood to that of other animals, typically seeking to feed during the daytime periods just after sunrise and just before sunset. It is a stealthy mosquito, often succeeding in taking blood meals from the lower body and ankles. The larvae are likewise evasive, descending to the bottom of their breeding containers due to changes in light or upon being physically disturbed. They remain motionless, often for a sufficient period of time to be overlooked. Principal identifying characteristics. 1. Small to medium-sized brownish-black species with silver accent markings. 2. Female abdomen is pointed. 3. Distinctive lyre-shaped pattern of white scales on the thorax. 4. Proboscis is unbanded, whereas tarsi possess pale basal bands. 5. Abdomen dark with narrow basal bands. Aedes albopictus, Asian tiger, or forest stay mosquito. Aedes albopictus is a species native to Southeast Asia. In recent decades, it has spread to Hawaii, North America, Central and South America, and Southern Europe. In the U.S., the species was first discovered in the area of Houston, Texas in 1985 and has since spread throughout the country. Scrap tires. Shiploads of natural rubber scrap tires have been identified as the source of Houston's initial population of A.E. albopictus. The interstate transportation of used and scrap tires is now the primary means of its dissemination. This species breeds abundantly in many of the same types of natural and artificial containers that A.E. aegypti is known to use, particularly tires. The Asian tiger mosquito can transmit several disease pathogens, e.g. dengue fever, yellow fever, and dog heartworm, to a limited extent. In the U.S., it has also been implicated as a potential vector in transmission of WNV, EEE, California encephalitis, CE, La Crosse, LAC, chikungunya, and possibly Zika viruses. However, most mosquito control personnel in the U.S. will agree that its aggressive biting behavior toward humans is the primary reason for most complaints. It seeks blood meals during the daytime about the same time as A.E. aegypti, 2 to 3 hours after sunrise and before sunset. The females are weak flyers, i.e. usually not dispersing more than several hundred feet from their larval development habitat. When they do fly, they generally stay close to the ground, therefore avoiding strong or gusty winds. There is substantial evidence that the Asian tiger mosquito, through competitive displacement, has replaced A.E. aegypti and A.E. triceratops, eastern treehole mosquito, in breeding sites in parts of their geographic range, except in large subtropical urban areas. A.E. albopictus has replaced A.E. aegypti in the U.S., for example, in Atlanta and areas north. A.E. aegypti is no longer found, however, in New Orleans, Houston, Miami, and areas south. A.E. aegypti is still present in significant numbers in some areas. Likewise, in more suburban areas where A.E. triceratops formerly was abundant, A.E. albopictus is now the primary artificial and natural container colonizer. Principal identifying characteristics. 1. Small to medium-sized black species with silver accent markings. 2. Female abdomen is pointed. 3. Similar in appearance to A.E. aegypti, but in place of lyre-shaped pattern, there is a distinctive median narrow strip of white scales on the thorax. 4. Proboscis is unbanded, whereas tarsi possess pale basal bands. 5. Abdomen dark with narrow basal bands. Aedes triceratops, eastern treehole mosquito. Although traditionally inhabiting woodland treeholes, A.E. triceratops can also often be found quite often with A.E. albopictus in the same ecological niche around human habitations in small urban and suburban areas. The species is frequently collected from man-made containers, such as discarded tires, buckets, and even beer and soft drink containers. When we talk about the fact that they can breed in even beer and soft drink containers, that's just making the point. It takes very little water for these mosquitoes to breed, so just keep in mind when you're inspecting a property for Aedes triceratops, they don't need much water to breed, and you're going to have to do a very thorough inspection. The bite is painful, and the species is frequently troublesome in both suburban and wooded areas. Aedes triceratops is considered the primary vector of CE and LAC in the Midwest and eastern U.S. It has also been implicated in WNV and dog heartworm transmission. Females tend to feed during the day but prefer shaded areas to full sun. Larval development is slower than that of A.E. aegypti and A.E. albopictus, and those species may predominate in areas where distribution overlaps during the warmest season. Aedes triceratops, however, is more cold hardy and ordinarily is most abundant early and late in the season. Principal identifying characteristics. 1. Medium-sized black species. 2. Female abdomen is pointed. 3. Tarsi and proboscis not ringed with light scales. 4. Sides of thorax covered with silvery white scales, separated by a broad central area of dark brown scales, which broadens apically and contains a small median light-colored area. 5. Abdomen blue-black with basal patches of lateral white scales. Permanent water mosquitoes lay egg rafts on the water's surface. Anopheles quadrimaculatus, malaria mosquito. Anopheles quadrimaculatus was historically and remains the potentially principal vector of malaria east of the Rocky Mountains. It has also been implicated in WNV and dog heartworm transmission. It occurs throughout the central and eastern U.S. and is very abundant in the southeastern states in rural and suburban areas. Anopheles quadrimaculatus is a freshwater mosquito found breeding in swamps, marshes, lake margins, rice fields, and slowly moving streams that are usually alkaline. The larvae prefer permanent or semi-permanent water containing floating debris or emergent vegetation, which provides protection from predators. During the summer, the larval period is relatively short, about 12 to 14 days. The pupal period generally requires another 2 to 3 days. Anywhere from 8 to 10 generations per year can be expected in most areas. Anopheles cruisens. Anopheles cruisens is susceptible to infection by malaria parasites in the laboratory. However, should the disease occur in the U.S., it is not regarded as a significant natural vector. It readily bites humans, and if sufficiently abundant, the species can become a significant pest. Anopheles crucians breeds in acidic, fresh water, such as that in cypress swamps, rice fields, and ponds. Larvae also occur in other habitats, such as lake margins and sluggish streams. Adults rest in shelters and are readily attracted to light traps. The flight range is about 1 mile. Principal identifying characteristics. 1. Medium-sized, dark brown species, palpi as long as the proboscis. 2. Female abdomen, dark brown and blunt. 3. Wings with alternating white and dark spots. Tip of wing white with 3 dark spots on anal vein, and front margin of wing dark scaled. 4. Palpi have white bands. Culex quinque fasciatus, southern house mosquito. This species is a major vector of WNV and St. Louis encephalitis SLE in Louisiana, and it has long been considered the primary vector of dog heartworm. While its preferred hosts are birds, it will readily feed upon humans, dogs, and other mammals. The abundance of the species and its feeding habits allow it to play a significant role in transmitting WNV and SLE from the bird reservoir to man. The species is most active at night and will readily enter houses to feed. The southern house mosquito breeds prolifically in water that contains a high level of organic matter. It occurs in roadside ditches, catch basins, septic ditches, sewage, oxidation ponds, stormwater pump station reservoirs, septic tank and sewage treatment plant effluent, water contaminated with waste from vegetables and meat processing plants, and in man-made containers. Somewhat paradoxically, the greatest numbers are often encountered during dry spells when evaporation exceeds replenishment of water, resulting in the concentration of pollution in water bodies as they shrink. Principal identifying characteristics. 1. Medium-sized light brown species. 2. Female abdomen blunt. 3. No distinctive markings on legs, proboscis, or wines. 4. Half-moon-shaped basal bands on abdominal segments. Culex selenarius. Culex selenarius reaches its greatest abundance in coastal regions. It is known as a vector of SLE and played a role in the transmission of the virus in the major epidemic of 1975 and in subsequent smaller outbreaks. More recently, it has been implicated as a likely bridge vector to bring WNV from birds to humans and other non-avian hosts. The species is most active during the spring and fall months when its populations attain their highest numbers. It is during the summer that CX, Salinarius larvae, and adults are difficult to locate. The larvae breed in fresh and brackish water in various habitats. Along the coast, breeding occurs mostly in the fresh, intermediate, and brackish marshes. The larvae may occur principally in either fresh or foul water in ponds, pools, ditches, and occasionally in barrels or bilge water in boats. The females bite readily outdoors, usually during the early evening hours, and occasionally enter dwellings. Principal identifying characteristics. 1. Medium brown, brownish species. 2. Female abdomen blunt. 3. No distinctive markings on legs, proboscis, or wings. 4. Abdomen mostly dark brown, scaled, often with narrow to moderately broad basal bands of dingy yellow scales. Culicida melanura. Culicida melanura is an important vector in the bird-to-bird cycle of EEE transmission. In this sense, they act to maintain both of these viruses in the local environment. CS melanura feeds exclusively on birds, consequently other mosquito species, such as Culex or Aedes species, are responsible for human and horse infections because they will feed on birds in addition to humans and horses. Little is known about the habits of CS melanura adults except that they, like many other species, are attracted to lights. Females can be collected from resting boxes in the morning hours by use of suction aspirators. Larvae of CS melanura often occur in small permanent to semi-permanent bodies of water, particularly around hardwood swamps and in boggy, palmetto-filled areas. The larvae occur during the late fall, winter, and early spring when water temperature is less than 60 degrees Fahrenheit. Culicida innerata, winter mosquito. Because it is readily attracted to light, it can be collected in significant quantities in light traps. The females are seldom persistent biters, although they do occasionally attack humans. The species prefers to feed on large domestic animals and may become very troublesome to livestock. CS internata has been implicated as a possible WNV vector. The larvae occur in such situations as ground pools, salt marshes, and ditches, often with pollution. Large populations occur in cattle hoof prints during the late fall, winter, and early spring when pastures contain abundant surface water. They have also been collected from abandoned tires and other artificial containers, as well as from abandoned swimming pools in the New Orleans area following Hurricane Katrina in 2005. Principal identifying characteristics: 1. Large species with speckled brown and white scales. 2. Broad wings. 3. Female abdomen blunt. 4. Abdomen with yellowish white dorsal bands which widen laterally. 5. Tuft of yellowish setae arising from the base of the subcostal vein on the undersurface of the wing. Coquilitidia perturbans, commonly known as the cattail mosquito. This is a troublesome species that bites in the early evening and occasionally during the daylight hours in shade near its breeding area. The females readily enter homes and bite viciously. They are strong flyers. EEE and WNV have been recorded from wild-caught CQ perturbans, but its role in the epidemiology of these viruses has not yet been determined. Species of the genus Coquilitidia have a unique morphological adaptation. They have a sharpened structure on the siphon tube of the larva and on the trumpets of the pupa. They get their oxygen by osmosis, and they stay underwater by using the teeth on the siphon tube to attach to plant roots. The aquatic stages, for the most part, are passed entirely submerged. Detection of breeding sites is very difficult as the larvae quickly detach themselves from the host plants whenever they are disturbed. This is also true for the species in the genus Mansonia. Breeding of CQ perturbans takes place in marshes, ponds, and lakes that have a thick growth of aquatic vegetation. Larvae have been found associated with alligator weed, pickerel weed, cattail, water lettuce, arrowhead, aquatic sedges, and other aquatic plants. Larval development is slow, usually requiring several months. Larvae, which are produced in one season ordinarily, do not complete their development until the following spring. The adults emerge in late spring or early summer. Throughout most of its range, CQ perturbans appears to have only one generation per year, though it may be possible to have more. Purpose and scope of mosquito inspections. Mosquito inspections are fundamental to understanding and controlling mosquito populations. By identifying breeding sites, population densities, and species presence, these inspections mitigate the health risk associated with mosquito-borne diseases such as West Nile virus, WNV, malaria, and dengue fever. The goals of mosquito inspections can generally be divided into two broad categories: surveys and surveillance. Mosquito control professionals conduct mosquito surveillance, remove places where mosquitoes lay eggs, control mosquito larvae and pupa, control adult mosquitoes, and monitor control efforts. Mosquito control efforts and capabilities vary across the United States. A survey involves a one-time collection of data aimed at assessing a specific mosquito problem or scenario. This could range from determining if mosquitoes are breeding in a particular container to evaluating a region for potential floodwater breeding. Surveys are typically more localized and focused, often conducted to assess an immediate issue or prepare for a targeted intervention. Surveillance, on the other hand, refers to an ongoing and systematic process of monitoring mosquito populations over time. The goal is to collect long-term data on mosquito activity, allowing for trend analysis, population modeling, and the assessment of control measures. Surveillance is crucial for understanding patterns in mosquito behavior, the spread of mosquito-borne diseases, and the timing of peak activity periods. Components of a mosquito control program. An effective mosquito control program relies on comprehensive inspections that gather critical information across multiple dimensions. A standard inspection framework typically includes the following. 1. Adult mosquito density and species composition. Knowing the number and types of mosquitoes present allows for targeted control measures. Some species are more likely to carry certain diseases, so accurate identification is crucial. 2. Rainfall and tide monitoring. Mosquito breeding is heavily influenced by environmental factors like rainfall, tides, and flooding. Regular monitoring can help predict breeding events. 3. Breeding site mapping, identifying and cataloging breeding locations such as stagnant water, artificial containers, or natural habitats is essential for proactive intervention. Advanced programs often extend their scope to include arbovirus monitoring in birds. Since birds can act as reservoirs for diseases like WNV, monitoring avian populations provides early warnings of virus activity. Arbovirus testing in mosquitoes. Mosquitoes collected through traps can be tested for viruses to determine the prevalence of a disease within a population. Specialized species surveys. These focus on locating specific vectors such as Aedes aegypti or AE albopictus, which are known carriers of diseases like Zika and chikungunya. Floodwater egg sampling. Sampling soil for floodwater mosquito eggs provides insight into potential outbreaks in areas prone to flooding. Initial versus ongoing inspections. Initial inspections often serve as a baseline assessment to determine whether mosquito control efforts are necessary. These one-time surveys focus on identifying hotspots or assessing the scope of the problem. Once control measures are implemented, ongoing inspections are essential to evaluate their effectiveness and adapt strategies as needed. For these efforts to be meaningful, consistency is key. Inspections must follow standardized methods and occur at regular intervals. Precise record keeping ensures that comparisons can be made over time and across different locations. Essential details for records include inspector's name, date of inspection, specific location, and details. I think there's a very high level of importance when we talk about these type of inspections. You have to be consistent. You need to do the same things on each inspection, and you do need to follow that standardized method because if everybody's doing something a little different, you're going to get different results. Also, you have to think about the fact of these occurring at regular intervals, so you need to have a plan on how the inspections are going to be conducted, where they're going to be conducted, and at what intervals, because if you don't do all those things together, again, you're going to get different records that are not really going to tell you exactly what's happening. As far as the details for the records, it is important to have the inspector's name, the date of inspection, and again the specific location and details. So if you don't have the date of the inspection, it's not going to be helpful. Most people, when you're searching for information, the first thing you look at is the date. If I don't know the date the inspection was done, that really doesn't give me any insight to what was going on or when it was going on or what the time was when it was going on. To streamline operations, maps with index grids are often used, enabling inspectors to mark and revisit specific sites easily. Digital tools like Geographic Information Systems (GIS) and GPS technology further enhance data accuracy, allowing for the integration of inspection data with broader environmental data sets. Egg surveillance methods and applications. Egg surveillance provides valuable insights into mosquito reproduction and breeding site distribution. Mosquito species vary in their egg-laying behaviors, necessitating different approaches to monitoring. 1. Ovitraps. Ovitraps are effective for monitoring container breeding mosquitoes, such as AE aegypti and AE albopictus. These traps consist of small jars or containers partially filled with water and lined with a textured material for egg laying, such as seed germination paper or velour strips. The strips are periodically collected and the eggs are counted under a microscope. Ovitraps are particularly useful for determining the presence or absence of species in an area. While they can provide indirect indication of adult population densities, their relationship between egg counts and adult activity is influenced by factors like seasonal cycles and generational overlaps. 2. Egg buckets. For permanent water species like culex, egg buckets serve as an effective tool. They are typically large containers filled with water enriched with organic matter, such as hay infusion or fish emulsion fertilizer. The organic material attracts females to lay their eggs, simulating natural breeding sites. Floodwater egg sampling. During droughts or dry seasons when larvae may not be present, soil sampling for floodwater mosquito eggs becomes a critical technique. Soil from suspected breeding areas is collected, sieved, and processed to extract eggs. By placing the soil in a saline solution, eggs float to the surface and can be easily identified. This method helps predict the potential for future outbreaks when flooding occurs. Larval surveillance. Larval surveillance. Larval surveillance is a direct method for identifying active breeding sites. Mosquito larvae, visible as small, wriggling organisms in standing water, indicate the presence of a breeding population. Without even asking the question, I'm going to say most of you have seen these larval wrigglers in the water. You may have seen them as a kid and didn't even know what they were. I know I did, and then as I started studying mosquitoes, I really realized every time I was looking in the water and I was seeing these little wrigglers, that was indicating the presence of mosquito larvae in the area. So sometimes something as simple as an eye test can let you know that there's mosquito larvae in the water that you're looking at. 1. Sampling techniques. Larval sampling is often conducted using a dipper, a shallow, cup-like tool designed for collecting water samples. Inspectors scoop water quickly, capturing larvae before they retreat to deeper layers. For containers or small water sources, syringes, turkey basters, or small nets are employed. The number of larvae collected per dip provides a quantifiable measure of breeding site productivity. Higher counts often indicate a need for immediate control measures. What we're talking about here with sampling can be really simple. You just have to be careful when you're trying to capture the larvae that they don't retreat down to those deeper layers before you get a chance to get some of them. Whether you're using turkey basters, nets, the syringes, it doesn't matter what you're using. The biggest problem I've run into is that they try to go down deeper. As noted here, one of the biggest reasons why we're doing these sampling techniques is to find out if we need immediate control measures. So that number of larvae collected per dip is really going to be quantifiable as we're talking about. It lets us know how bad things are, how many mosquitoes are breeding in the area, and do we need those immediate control measures. 2. Environmental considerations. Understanding the habitat preferences of different mosquito species enhances larval surveillance efforts. For example, floodwater species rely on temporary pools created by heavy rains or tidal flooding, while permanent water species favor stagnant ponds or artificial containers. Timing inspections to align with environmental triggers, such as recent rainfall or irrigation, increases the likelihood of identifying active sites. So if we really think about it, we've got so many tools nowadays that can really help us when it comes to mosquito monitoring. It's almost unbelievable. Here you're seeing the U.S. Drought Monitor. This is a website anybody can go to, but the reality is I can look at the U.S. Drought Monitor and I can get an idea of where my area is at. Or even if I'm going to be doing mosquito control in an area I'm not familiar with, I can get an idea if they're in a drought or not. Now obviously putting your eyes on the area is most important, but I think using the tools provided to us nowadays via things like the internet is something we need to take advantage of. I mean, think about the weather. We can easily look at the 10-day forecast and we can get an idea of what our weather is going to be in the future. Now, I'm not going to tell you the weather's always correct, but the point is, if we looked at past knowledge with the weather and we look at what's coming in the future, it can help us figure out when to time and what to look for with our larval samplings for mosquitoes. Check out this use of technology. In this instance, I'm using Google Maps to look at the area around where I live. I'm looking and just thinking about it. I live in an area where we've got a lot of rivers with fresh water mixing with salt water, the Gulf of Mexico. I've got a lot of marshes, I got a lot of mangroves. I've got some really interesting areas for mosquitoes to breed. So just think about using a tool like Google Maps and what that can show us about our area and give us insight on where we would need to do larval samplings for mosquitoes. I wanted to show you another scenario where we could use a tool like Google Maps. In this instance, we're going to use the measuring tool on Google Maps to look and see from an overview, from like a satellite overview, how far away the nearest water source is. So as you're seeing on here, we're measuring the distance from the home we're at to the nearest water source. Now another thing you're able to do as well is you can measure where the nearest wooded area is, or whatever the case may be. Wherever you think there could be an issue with mosquitoes breeding, you can measure from the house you're at. If you understand the different species, then you would be able to understand if that species could travel that far to the home or property where you're doing the treatment or sampling at. Adult mosquito surveillance. Monitoring adult mosquitoes provides critical information about species composition, population dynamics, and disease risk. Adult surveillance methods include both active collection techniques and passive observation. 1. Landing count rates. In this method, the inspector acts as live bait, counting the number of mosquitoes landing on exposed skin within a set time frame, typically 1 minute. While simple and cost-effective, this approach requires caution due to the risk of bites and potential disease exposure. And as you can see on the screen here, I've always found this to be a very interesting method. I personally don't want to go out and get eaten up by a bunch of mosquitoes just to see what a landing count rate is. I will say this, I know a lot of people in the industry and especially at the universities and things that are doing it, and it is very effective. Not something I want to do, but at least you understand it can be an effective way to see what's going on out there. 2. Mechanical traps. Mechanical traps are a cornerstone of adult mosquito surveillance. They are designed to attract and capture mosquitoes for identification and disease testing. Common trap types include New Jersey light trap, a classic device that uses light and suction to collect mosquitoes, ideal for general surveillance. The New Jersey light trap was developed in 1927 at the New Jersey Agricultural Experiment Station. It has been used by mosquito control agencies and Rutgers University since 1934 for mosquito research and planning mosquito control activities. The trap was designed to maximize adult surveillance results and minimize human labor and bias. So the bottom line with this trap is it was actually developed because we didn't want to have all the labor that goes into going out and looking for these adult mosquitoes, and also the bias, because as we talked about, even if you think there is a standardized practice or set of standards people use, everybody is going to do things a little bit different. So this New Jersey light trap was great because you put it out, it does its job, you go back, you collect it after X number of hours, and you don't have to worry about as much human layer and or the bias involved. When we look at the gravid trap, it's designed to attract females ready to lay eggs. This trap uses odorous water as a bait and is especially valuable for arbovirus monitoring. The gravid trap is a very interesting tool in my opinion. Obviously you can see it was designed to attract females ready to lay eggs. It was also designed to collect Culex mosquitoes. It was used a lot in the Memphis, Tennessee area, all the way down to New Orleans along the Mississippi River, because there was a lot of Culex mosquitoes in the area, and they especially wanted to start monitoring for that arbovirus and just making sure of what was going on in the area. So the gravid trap was really useful in this arena. Also, it's something you can use nowadays. I would definitely look into something like this, especially if you're doing mosquito control on any grand scale. CDC and EVS light traps. These versatile traps combine light and carbon dioxide to attract a broad range of mosquito species, including those active during the day. When talking about CDC and EVS light traps, it's important to note that mosquito species include a wide range of mosquito genera and species, including Aedes, Anopheles, and Culex, and the life stage we're going to attract is the adult. When we look at tips for use for a CDC or EVS light trap, you're going to hang them from a tree about 5 feet above the ground. You're going to use dry ice, releasing carbon dioxide that attracts the host-seeking female mosquitoes. They're most effective when set at dusk and collected after dawn the next day. In this image provided by the CDC, we're seeing a CDC light trap capturing flying mosquitoes. You can see exactly how it hangs from the tree, and as we mentioned before, it needs to be about 5 feet above the ground. Next, we're going to take a look at rotator traps. By collecting mosquitoes hourly, these traps provide insights into daily activity patterns, aiding in the scheduling of control measures. And I think these are very interesting because we are collecting samples of when the mosquitoes are most active hourly. So this provides those insights that we need to see their activity patterns, when they're going to be most active, so we can schedule control measures correctly. Understanding that's a big thing. Now, if we understand the different types of species, we also understand the times of day they're active, but this is just another tool for us to use to get more insight in those activity patterns. Mosquito rotator traps work by mimicking an animal or person, sending out signals that mosquitoes use to find their prey. The main signal is a plume of carbon dioxide, CO2, as if an animal is exhaling. Some traps send off a steady amount, while others match the rhythm of breathing. These mosquito rotator traps are very interesting to me. I love the idea of them emitting carbon dioxide to attract female mosquitoes. What better way to bring the female mosquitoes in? So the concept is really a great idea. Now, obviously, it's like an animal is exhaling basically at that point, so we're fooling those mosquitoes pretty well. But then when we look at the fact that they can match the rhythm of breathing, that's a whole nother concept, because now we're really fooling those mosquitoes to come in. So once again, I think these are great on those larger mosquito control situations, more of a grand scale. You can use them on smaller properties as well. I just think this is a really good tool, some good technology, and I would definitely start to incorporate it in your mosquito control situations. BG Sentinel Trap, specifically designed for AE aegypti and AE albopictus. This trap uses human scent lures and CO2 for high efficiency sampling. With BG Sentinel mosquito traps, we're going to be collecting the life stage of the adult. Tips for use: use an attractant, e.g. octanol lure, human scent lure, carbon dioxide to attract more female mosquitoes. So as you're seeing here, we're using some of these same attractants in each type of these traps, and again, the goal is to get as many female mosquitoes as possible in the given area. And again, talking about BG Sentinel traps, obviously we talked about before, they use an attractant to attract more female mosquitoes, and this is an image of a BG Sentinel trap provided by the CDC, so this gives you an idea of what they look like. 3. Resting collections. Many mosquito species rest in shaded, humid environments during the day. Collecting mosquitoes from these resting sites, such as hollow trees, culverts, or artificial shelters, provides valuable data on species that may not be active during trap hours. Resting traps are used for mosquito species Anopheles, Culex, and Culicida. With these traps, we're targeting adult females. When looking at tips for use with resting traps, we're going to set traps in cool, shaded areas, collect mosquitoes in the early morning. Do not set in areas with high sun exposure for extended periods of time. On the current slide, you're seeing an image of a resting trap. A resting trap is best used for catching mosquitoes by placing in cool, shaded areas, as we mentioned before. This photo is courtesy of the CDC to show you what a resting trap looks like. Alternatives to dry ice used in mosquito traps for surveillance. For vector surveillance, many programs use dry ice baited traps to collect host-seeking mosquitoes. Commonly used traps include CDC light traps, baited with both dry ice and a light source, and BG Sentinel traps baited with both BG lure and dry ice. Dry ice serves as an inexpensive and convenient source of carbon dioxide, CO2, to attract mosquitoes. In case of a dry ice shortage, consider other options. Traps can be used with their originally designed lures, e.g. BG Sentinel traps using the BG lure only, CDC light traps using light only. Aedes aegypti and AE albopictus may not be efficiently captured without CO2 or another attractant. CO2 tanks provide the best alternative source of CO2. Use of CO2 tanks should be balanced with increased program cost and logistical considerations. Yeast fermentation 1-2 could provide a temporary alternative CO2 source if dry ice is not available. Yeast fermentation produces lower levels of CO2 than dry ice. Traps baited in this way capture fewer total numbers of mosquitoes, though species diversity appears similar. Methodology should be validated by programs for their surveillance targets prior to broad use. Overcoming challenges in mosquito surveillance. Mosquito surveillance programs face several challenges, including species-specific behaviors, environmental variability, and the need for consistent methodologies. Factors like weather conditions, habitat changes, and urbanization can impact mosquito activity, requiring inspectors to adapt their strategies. Best practices include training. Inspectors must be well versed in mosquito biology, species identification, and habitat preferences. Data standardization. Using consistent forms, tools, and techniques ensures reliable comparisons over time. Technological integration, employing GPS, GIS, and real-time data systems, streamlines operations and enhances accuracy. Conclusion: effective mosquito inspections are the foundation of any mosquito control program. Before conducting mosquito control, it is important to know the biology of mosquitoes and to know specifically which mosquitoes are present. Therefore, mosquito identification is one of the crucial steps in identifying risks for both biting mosquitoes and mosquito-borne diseases in an area. By combining detailed surveys, continuous surveillance, and advanced methodologies, these inspections enable targeted interventions that protect communities from the risk posed by mosquito-borne diseases. As new challenges emerge, the adaptability of these programs ensures their ongoing relevance and success. The use of trade names in this publication is solely for the purpose of providing specific information or as examples. American Pest CEUs does not guarantee or warranty the product's name, and references to them in this publication do not signify our approval to the exclusion of other products of suitable composition. All chemicals should be used in accordance with directions on the manufacturer's label. Use pesticide safely. Read and follow directions on the manufacturer's label. We thank the photographers that allowed us the use of their great images to enhance the course content. Let's take a quick moment to acknowledge the contributors of content and images for this course. Congratulations, you have completed the course. Please proceed to the quiz.